the patterns of movements that we have affect how the diseases can spread. And what if movement generates some sort of heterogeneous agglomerates, okay? So if instead of having everyone well-mixed, distributed, homogeneously distributed in space, what happens if there are clusters, okay? This is the kind of questions that we are interested. And as we are going to see, active motion, and I will explain what that means, breaks the well-mixed hypothesis, and we get new situations. So this project tries to put together active matter and epidemics. This is not the first time that people started to do this, but this is a recent field. We know that self-propelled organisms, they can agglomerate, even us, okay? So we move to the city center to find something there, because there is a high density of people selling something that you are interested in, for instance, okay? So our movements and the movement of organisms in general, they are self-propelled, and they depend on the density of other organisms, okay, due to social interactions and things like that, okay? And we know, as I said, from the pandemics, that agglomeration affects the spread of infections, okay? And here, when I say infection, actually I would like you to generalize this idea. What I mean is some sort of internal state which is transmitted by close contact, okay? Some information that is inside the individual and that by contact it is transmitting to that information to the other individual, okay? So we hope that combining these ideas can be relevant to study epidemics themselves, but also information spread in colonies of insects, for instance, and also phage therapy. Phage therapy is a quite interesting scenario where you are going to use viruses to kill bacteria, okay? So you know that viruses are much smaller than bacteria, and they move through diffusion, okay, of the environment, whereas bacteria, they move actively, so they swim, okay? That's completely different. Now imagine that you have a collection of bacteria. This collection of bacteria is distributed in space in a heterogeneous way, and then you start to spread a virus from bacterium to bacterium, okay? This can be relevant because we know that bacterial infections, they are developing some sort of resistance against antibiotics, okay? So this is a new kind of therapy called phage therapy. It's actually not new. Since the Chinese, there was something along those lines. But the point here is that they are trying to understand more and more how virus and these kind of ideas can spread between bacterial colonies, okay? So it can have this kind of application as well. So the general goal of this presentation, of our work here, is to develop a simple framework to study the relation between agglomeration and transmission, okay? Mediated by active motion in this case, and I will explain what that means. This is a work in collaboration with Guilherme Costa, who is here, presented earlier today. Okay, so what is active matter? I'm happy that yesterday Ignacio actually introduced these very well. So active matter is a class of non-equilibrium statistical mechanics systems where each particle has self-propulsion. So instead of being just a Brownian particle subject to fluctuations of the environment, it actually has self-propulsion. So it can swim or move in a persistent manner. So examples include animals like fish, birds, sheep, and also ants, people, cells, microfilaments, artificial colloids as well. So there are many situations. And in this case, these systems are non-equilibrium systems in which energy is injected and dissipated at the microscopic scale, at the scale of the particle size, okay? Giving to each constituent an irreversible dynamics, okay? So what is the basic unit of active matter? It's basically the free self-propelled particle. So it's a self-propelled particle that does not interact with any other particle. So one example of active particles in general is E. coli bacteria, which is going to swim by beating flagella. And the kind of movement that we see is this. This is called run and tumble motion. The particle will move in a straight line at a certain speed, And at a certain moment, which is stochastic, it will completely reorient its trajectory. Okay, so you get this kind of pattern. So you have two parameters in the model, the speed, v, and the rate of reorientation. Okay, so this is a stochastic, right? The process here is a Poisson process that decides if the particle will change or not the direction. Okay, and with these two parameters, you can define what's called persistence length. This is quite important. If the persistence length is small, essentially you have a Brownian particle, the usual Brownian particle, because the particle is changing directions all the time. Okay? Imagine that you have a pollen particle, and it is receiving kicks from the environment, like water molecules. Every time it will go on a different direction. Okay? So this is a normal Brownian particle. Persistence length is almost zero. Okay? But for bacteria, this is not the case. It will move in a certain direction for some time, then it will change directions, so on and so forth. Okay? So alpha is smaller, the rate of reorientation is smaller, and therefore the persistence length is larger, which is the average of these runs here. Okay? And this will give completely different phenomenology for active matter as compared to passive matter. So one of the examples is this. Here we have an experimental setup of active colloids which move in a similar fashion, so to speak, compared to bacteria. And what you have is persistent motion. These particles, they move persistently, and steric interactions, meaning that these particles cannot penetrate into each other. Okay? So they are stiff particles. And what you get is spontaneous agglomeration. Okay? So this is the phenomenon that was described yesterday by Ignacio of motility-induced phase separation. Okay? You start with a homogeneous distribution of particles, and spontaneously the system will start to form these clusters. Okay? So how is that possible? If there are no interactions in the sense of attractive forces between these particles. Okay? There are no attractive forces, and yet we see this kind of patterns emerging. So the explanation here is basically that when these particles meet each other, they keep trying to go in the same direction. Okay? So they stay bound to one another. So effectively, this is like an attraction. Okay? So persistent movement generates an effective attraction between the particles. Okay? And if the persistence is high enough, what you're going to see is that a third particle can come, and you have now a cluster of 3 particles. Then the larger the cluster, the more likely it is hit by a new particle, and then the sizes of the clusters will increase exponentially until you have some saturation, because the number of particles outside the clusters is small, and the rate at which particles join clusters is equal to the rate at which particles escape from clusters. So you have a dynamical balance equilibrium between these, generating a distribution of cluster sizes in the steady state. So you have the stationary distribution of cluster sizes. And this is completely unique to active matter in the sense that you have formation of clusters without any attractive forces between the particles. Okay? So this is the kind of model that we're going to study. We are going to have particles on a lattice. They will form clusters, and we are going to spread diseases between these particles. I'm not saying that organisms are going to form clusters using the same mechanism that I'm presenting here. This is only one simple way that we are implementing to have clusters and study what is the role of clusters and these kind of heterogeneous spatial distributions in spreading of diseases. Okay? So in this model, we have a lattice. Each side of the lattice can have at most 1 particle. We have a dimensionless global concentration phi, which is the fraction of particles in our lattice. Each particle has 4 possible directions. Okay, it's a square lattice in 2D. Particles are selected sequentially at random. And then I select 1 particle. If this particle has a self-propulsion that points to an empty site, it will just go to the empty site. Okay? It will keep moving on that direction. If not, it will be blocked. Okay? This is the steric part of the ingredients. And at a fixed rate, every time that you choose a new particle, there is a certain probability alpha that this particle will change its internal direction of self-propulsion, okay? So we have these two parameters, as I said before, the speed at which it moves. This is basically 1. We're going to set the speed to 1. And then there is the tumbling rate, which is our parameter to control the persistent movement of the particles. And we have periodic moderate conditions, random initial states in positions, and self-propulsion, okay? So this is the kind of picture that we have in 1D, okay? I just showed you the model in 2D, but actually I'm going to show you this picture from this paper in 1D, where things are easier to explain. So basically, first you have the formation of the clusters, okay? So you have these orange clusters here. They are orange because these particles are susceptible. Okay, so orange is susceptible, red is infected, and green is recovered. We are going to have an SIR kind of model here for the spreading. So initially, all of the particles are susceptible except for one. Okay, so one of the particles in red is actually infected. And another thing that we see at the beginning, so by the way, time is here on the y-axis, okay, and position is here on the x-axis. So one thing that we see in the initial state, and actually in all subsequent states, is that there is a certain characteristic distance between the clusters, LG, and a certain size for the clusters, LC. Okay? This is an spontaneous thing that emerges from the level of persistence and from the density of the global system. Okay? And these particles that are infected, they will start to infect others at a certain rate, lambda, provided that these other particles are in direct contact with the infected particle. Okay, so it's infection by direct contact. Once the particle is infected, you can have that at a certain probability beta, the particle will actually recover. Okay, so what's the type of dynamics that we see? Well, here you can see that first, the initial cluster gets infected, then particles which are infected at the border of the cluster will flip and take the disease to the next cluster, and so on and so forth. So clusters will get sequentially infected. Okay, so this is what I just explained. We are going to repeat these until the epidemic ends. Okay, and this is the kind of dynamics that we have. Here in the y-axis, we have the population fraction of a given type, infected, susceptible, or recovered. Okay, and here time since the first infection. And what we see is a sequence of peaks. Okay, so this sequence of peaks is basically what happens every time that a new cluster gets infected. Okay, so the number of particles that are infected starts to decrease exponentially because of the recovery, and then one particle that is infected reaches a new cluster, and then you have a new peak of infections, and so on and so forth. Okay, so there is a sequence of peaks. Eventually, particles will recover before they reach the next cluster. Okay, and these will mean an absorbing state where you have no cluster infected particles anymore. Okay, so as I said, when the infection reaches a particle at the border of a cluster, two scenarios are possible. One, the particle will flip, move away, and infect another cluster, or the particle will recover before that. Okay? So when this happens, is the end of the epidemics. So will the particle remain infected over this time scale? So the persistent movement of the particles and the density of particles, both of these things are going to set the typical distance between the clusters, okay? And the persistent parameters of the particle will set whether or not the particle will be able to cross this distance between the clusters without getting recovered, okay? So by answering these, we are actually answering when the epidemics on average is going to finish, and therefore how many people get infected. So our goal is to understand the fraction of infected individuals integrated over time. Okay? So we take all time instants, we sum the amount of people that get infected, and this is our order parameter divided by the total number of particles. Okay? We are going to call this FR infinity. Why R if we are talking about infected? Well, R because when the epidemics finish, what happens is that all particles that are recovered in the end, they are the ones that were once infected, okay? So by getting the number of recovered individuals at the end, we are actually getting the time-integrated number of infected individuals, okay? Okay, so now, here is an important question. Is it possible to have macroscopic epidemics? So what is a macroscopic epidemic? A macroscopic epidemic is a situation where you keep increasing the system size, okay? So you do new experiments with larger and larger system sizes, and the amount of infected individuals grows with the system size, okay? So for instance, you have an epidemic situation with 1 million individuals, and let's say that only 1,000 got infected. Now you're going to do the same thing with 2 million individuals, and if we have a macroscopic epidemics, you expect something like 2,000 individuals to get infected. So it is a real epidemic in the sense that no matter the size of the system, you are going to get a significant amount of individuals infected there. It could be possible to have a situation where in the thermodynamic limit, where you have an infinite number of individuals, the fraction of individuals that gets infected is actually so small that it goes to 0, okay? So this is the question. Is it possible to have a macroscopic epidemics in the 1D version of the model that I just showed you? And the answer is no. You cannot have a macroscopic epidemics. You can still have an epidemics as a finite size effect. So you're going to have a certain number, certain fraction of individuals that gets infected. But in the thermodynamic limit, this goes to 0, okay? And the reason is quite simple, actually. So imagine the best possible situation for the epidemics, okay? Which is all sites occupied. So you have that all sites are occupied, phi is equal to 1, okay? So the fraction of occupied sites is 1. In this case, there is always a probability that the next particle, so suppose that I am an infected particle, and sitting next to me is a susceptible one, there is always a probability that I'm not going to infect the particle sitting next to me because I'm going to recover first, okay? If this probability is finite, it will happen eventually, okay? And when this happens, there is still an infinite amount of particles to infect, okay? So if there is still an infinite amount of particles to infect, the fraction goes to 0. Okay? So there is no macroscopic epidemics in 1D, okay? It's still interesting as a finite size effect, but there is no macroscopic epidemics. Okay, so what happens in 2D? This is what we are actually doing now. That's the work in progress that I mentioned with Guilherme here. So things are different in 2D, okay? First of all, let me compare two situations. I'm going to have one case where V is equal to 10 and alpha is equal to 0.1, and the other V, the velocity, is equal to 50 and alpha is equal to 0.5. As you can see, in both situations, the persistence length is the same, okay? And when the persistence length is the same, you're going to have the same distribution of cluster sizes, okay? The same sizes for the clusters on average. And you're going to have the same distribution of contact degrees. So if you count the number of particles that I am touching, you're going to have a certain number of contacts. If you do a distribution of contact degrees, which is the name of this quantity, you're going to have the same thing in both situations, okay? So you have two situations that have the same distribution of contact degrees, and yet, as you are going to see, on the left, the epidemics stays contained within the initial cluster, and on the right, the epidemics conquers the entire system, okay? So what's going on here? Okay, basically what happens is that the travel time of the particles between the clusters is different. This is because the travel time of these particles are going to depend on something called active diffusion, okay? So these particles, they are going to move ballistically for some time, called the persistence time, but eventually they will start to reorient, okay? So if you look from afar, or in very long time scales, what you're going to see is that there is some sort of normal diffusion behavior in the long time limit, okay? And when you calculate this kind of normal diffusion behavior, the diffusivity goes as v squared over alpha, okay? So what this tells you is that this diffusivity will control the travel time of the particles between the clusters, and this travel time will decide whether the particle recovers before reaching the next cluster or not, okay? And here we have v squared over alpha. So even though in the two cases we have the same v over alpha, we do not have the same travel time, okay? And therefore, we do not have the same epidemic outcome, okay? So this is showing how the travel time can actually decide on the epidemics. And in fact, in these two situations here, these two curves, we have the same static contact network of degrees, okay? And yet, we have completely different situations in terms of the epidemics. Here, the number of infected individuals undergoes a second-order transition as we increase lambda, the infection rate, probability, if you want. And here we do not. Actually, this will happen here for large lambda, but you can see that the critical lambda is completely different, even though we have the same static network of contacts, okay? This is important because usually in epidemiology, people have a certain network, they decide, they impose the network, and they study what is the outcome of the epidemics. Here, we do not impose the network, but even in situations where they are the same, the result is different because of the dynamics between the clusters, okay? So here what we have is FR infinity as a function of alpha, our tumbling rate, okay? If alpha is too large, the clusters will be small and they will break apart very easily. This will make the disease escape from the clusters, but the clusters are so small, so the number of infected individuals is not so large. If we have a small alpha, the clusters will be really large, but they will be static in the sense that it will take a long time for a particle to flip at the interface of the cluster and go to the next cluster. So the number of infected individuals is also smaller. In between, there is a sweet spot where we have an optimal infection or number of infected individuals, okay? Just by controlling the persistence of these particles, okay? So now comes the question. Is it possible to have macroscopic epidemics in 2D? And now the answer will be different. Otherwise, I would not be saying it like that. In 2D, there could be site percolation. So what is site percolation? Site percolation is a situation where you occupy your lattice with a certain probability alpha, phi, which is the density of particles, and you find a path connecting one side of the system to the other side of the system just by going from particle to particle in their neighborhood, so by going through neighboring particles, okay? So you can ask what happens in terms of percolation here, okay? You can have a contagion percolation or you can have a geometric percolation only in this system, okay? So what we find is that even if our phi is smaller than the percolation transition phi, which is 0.59 in this case, we can still have contagion percolation, okay? So the particles are not going to be touching each other in a way that makes possible for the disease to spread only geometrically through the whole system, but we are still going to have a macroscopic epidemics and therefore a contagion percolation, so to speak, because of the movement of the particles which promote contacts across time between these particles. So activity here induces an effective contact network which has more contacts and connection pathways between the clusters and the particles, effectively bridging them together across time. So even though particles are not touching each other in a way that allows for percolation, if you wait enough, they will move and enter in contact with the other particles, and this will allow for a contagion percolation. So here we do have a real macroscopic epidemics. In fact, here I just showed you that for two different system sizes, we have that these two curves collapse. Okay, so if we keep increasing the size of the system, the amount of particles that are infected also increases, and effectively, what you have is a macroscopic epidemics here, okay? And this is because if the infection dies in one place, there will be some other path of infection that will continue the thing for you, okay? This is not possible in 1D, okay? So what's the role of dimensionality here? In 1D, even at percolation density, which is 100% in 1D, there is no macroscopic epidemics. In 2D, even if you are smaller, at densities smaller than percolation, activity can induce a contagion percolation, and therefore a macroscopic epidemics, okay? So the preliminary results that we have here is that the total final amount of infected individuals undergoes a continuous transition. The average contact network is not sufficient to determine the epidemic outcome, and this indicates that there is some sort of effective temporal contact network generated by the active dynamics, which needs to be considered in order to really determine if there is percolation, effective percolation or not, okay? And in fact, when we take the time-integrated contact network between the two situations of low persistence and high persistence, we can see that they give completely different contact networks, which can explain what we are seeing here. This is something that we are starting to touch at this moment. And just an extra before I finish, we are now also studying the CIS case, where instead of susceptible, infected, and recovered, we have susceptible, infected, and then back to susceptible, okay? So there is this possibility. What we see first is that these clusters start to get infected, okay? The infection kind of oscillates in space and time inside the clusters, and it starts to spread from cluster to cluster in a slow fashion, okay? And so eventually it will take over the entire system. It's kind of boring if you compare to other situations, okay? Now, what we're going to do is the following. We're going to introduce a new ingredient, which we call panic, okay? So what is panic? Imagine that the particle realizes that someone is infected in their neighborhood, okay? In this scenario, it will increase the tumbling rate, okay? So with panic, this is what we see. So essentially, the clusters here, they start to break apart, okay? This is basically because the particles are infected, and they are making the other particles tumble more. The persistence is going down, and the persistence is what makes the particles clusterize, okay? Once they are outside the clusters, they go back to their normal no panic state, and they can clusterize again until the epidemics comes back, okay? And in fact, if we plot the two situations with panic and without panic, we see that without panic, the number of infected individuals grows slowly by taking over more and more clusters, and then it oscillates, okay? But with panic, what we see is that initially, panic is worse, okay? Why initially panic is worse? Because if you are making the clusters break apart, because you are less persistent now, you are also allowing the disease to escape from the clusters, okay? So initially, you allow the disease to escape from the clusters just by not being persistent enough. But then the clusters are not formed anymore, and this makes the amount of infections go down, okay? So in the long run, it is good for these parameters to have this panic strategy, okay? Okay, so the take-home message is essentially that active particles can agglomerate spontaneously and generate dynamic contact networks. And these structures significantly impact epidemics between active particles. And we hope to use these ideas to connect to epidemics, information spread, and even phage therapy in the future. So a little commercial break. So this is the Brazilian workshop on soft matter here at ICTP-SAIFR, which we are organizing. And the registration deadline is February 15th. It will happen in April. If you know anyone interested, please let them know. These are the collaborators, and I'm happy to take any questions. Thank you.
Okay. Questions? Thank you, Pablo. I have a question about the when you changed from SIR to SIS. The SIS, at least in phi equal to 1, is a one-dimensional contact process, no? And it has critical value for to have like percolation of the disease. So my question is, have you checked in the SIS if you can have macroscopic epidemics in 1D as well?
That's a good question. I didn't think about these. Actually, the SIS thing started 3 days ago, basically, when we were discussing Guilherme and I. So these are really, really fresh results. But it's a very good question which we have to put on our to-do list.
Yeah, I mean, I don't know. I thought it could be interesting to see how different propagation process, in general, propagation processes change the dimensionality of the...
Yes.
I don't know.
Yes, this is the kind of research program that we have here. We want to explore very different ingredients for these guys, combining space and disease spreading. Yeah.
Yeah, you have emphasized the importance of the kinetics in determining when it's infecting. Do you have an idea or an estimate of which is the relevant dimensionless parameters that will control competition between motion, disassembly of clusters, and infection?
Yeah, so basically we have to have in mind persistence length and the rate of infection divided by the rate of recovery. These are the two main dimensionless parameters that we have in our system. But what we have actually is a non-monotonic behavior, right? So when we change these parameters, eventually we're going to have some peak of infections, an optimal behavior, because of this balance between having too many clusters or too many clusterized particles, because the particles are really persistent, against the other situation where eventually everyone is moving everywhere. So yeah, there is this balance between the two situations, and the main parameters are persistence length and in fact, the ratio of infection rates.
Thank you. Incredible work you have there. And I have 2 questions. The first one is, why don't you consider mortality rate? Because you're considering a recovery rate. And if you consider mortality, that if you think as that particle being susceptible to mortality, it will get off this dynamic, essentially, because it could not go on infecting other clusters and stuff. And the second question, I think there's an incredible work you have there, and I mean, like, since each year we have more ongoing diseases and outbreaks, I was going to ask, since we had a lot of discussion on whether to do or not lockdown, especially here in São Paulo and other small big cities, I would ask you, especially seeing your work with panic and not panic, because I can look at, really looks like all the discussion we had. You believe that on big cities, at least, I mean, like, if you were asked to contribute as a physicist, as an academic, on this decision making, what would you do? That's it.
Okay, that's very good 2 questions. The second one is actually, my answer is, please, policymakers, do not listen to me, because this does not translate directly to what's going on in the real world epidemics of humans. But I get your point. This provides insights into the level of clustering and how these affects epidemics. I agree with you. What I can say from our results is that there is a non-monotonic behavior. There can be some level of clusterization which is worse or better for the disease to spread, right? So in principle, there are situations where doing a lockdown would be not good, but of course, there are many other things at play here, and I don't want to say that this is what should be done. The other question, the first one about death, right? Yes. So we are assuming here that the time scales are such that the particles do not die. Okay, so basically everything is happening before there is sufficient time for them to die. But if we are considering a phage therapy scenario where viruses are killing bacteria, then this occurs much faster. And then it would be interesting to consider death. In this case, there are two possibilities. Either the particle that dies becomes food for the other bacteria, or it just stays there, sort of blocking their movement, okay? So depending on the kind of situation that we have, and what we do with this particle that dies, we can have different dynamics.
Okay, so let's think. Oh, one more question.
Do you intend to consider SIRS models? Because these are somewhat closer to what we see in endemic diseases.
Yes, this is something that could be done as well. Actually, there are, as I mentioned before, many possibilities for this research program. Yes, this would be one of the possibilities as well. I don't really have a feeling right now of what would be the main differences that we would see there, but probably we would see differences, especially comparing the situation with and without clusterization or spatial dynamics, which is the novelty here.
Okay, so thank you, Pablo.
Thank you.
Now, Davi.